Hello class, my name is Mulcahey and today I am presenting my microcosm project presentation for EBSP 416, which is General Ecology for American Public University. So this assignment was undertaken by me as a way to test the natural ecosystems in the desert biome that I live in. So hoping to learn how the ecology of the Ambergris River existed if the water were to stop moving, as in instead of moving forward, there were just small pools of water left where the Ambergris River would have been. I was curious as to what would survive in those little pools. I already had a base knowledge of ecology. Spent half of my years every year studying ecosystem in the Spring Mountain Range. I studied plant growth as it relates to the environment, the effects of too little precipitation, and this last year, the effect of too much precipitation, which I've always thought was interesting because it directly relates to the population and the numbers of the elk and the mule deer up in the mountain range. I took this class to further my knowledge of what I already do as a living and as a hobby, and to better present my data in scientific form. Studying microcosms helps us to learn how ecosystems work. In some cases, microcosms can help us determine how a toxic substance affects an ecosystem. This presentation is a representation of an underground river ecosystem when it surfaces. I plan to cover microinvertebrates that are found in this area of the river, as well as some of the plants. I made a simple hypothesis for this experiment that if I placed a young cattail in my microcosm, they would outlive a sample of algae, which I believe is Natrium desmid, but it's hard to say because most of the algae in this area look the same. These two pictures represent the area of collection, which is just south of Beatty, Nevada. This is part of the Ambergris River. It surfaces north of Beatty, travels through Beatty, comes down the side of the highway, and eventually finds its way back underground, right past where my collection was. For the collection of my water and soil from the collection site, I used a 4-ounce measuring cup. I first collected a 4-ounce sample of the soil from below the surface of the water. After the soil sample was collected, I rinsed the collection vessel out and collected 8 ounces of water without touching the soil under the water. Once my soil and water were collected, I proceeded to collect my autotrophs. These consist of a young cattail and a small selection of algae, which again I believe is Natrium desmid. My microcosm project ran for 7 weeks, received an average of 3 hours of indirect sunlight and 1 hour of direct sunlight each evening, and this is due to the shade sail behind the house, and the 1 hour of indirect sunlight is between where the sun reaches past the shade sail and above the trees. The water temperature inside my vessel stayed an average constant of 68 degrees per the thermometer on the side of the jar. By week 4, my hypothesis had been proven wrong. My cattail did not outlive the algae. The algae had grown to fill almost the whole water column. The heterotrophs held out for a long time, and in week 5 I had a damselfly hatch. This heterotroph lasted 2 days before expiring. The following slide contains a graph that represents the 4 major observation types of the 7-week period. In the chart you can see that by week 4 my cattail died, and in week 6 all heterotroph life had expired. The chart shows that the algae had bloomed and grown very well into week 5, where it started to die off at the soil level and started to decompose and lift towards the top. What the chart does not show is that at the soil level starting in week 6, there was new algae growth, which was bright green. So here's the chart I was talking about. The numbers on the y-axis represent the level of growth and heterotrophs and water clarity for representing a very high amount, 3 represents a high amount, 2 a medium amount, and 1 represents a low amount. For the weeks 4 through 7, where there's no data present, it represents a part of the microcosm it crashed. And in 4 and 5, the cattail is where the data was non-existent. And then for week 6 and 7, the heterotrophs had joined the cattail and expired. In this slide, I have 3 of the heterotrophs that I had been able to sample and get pictures of before sealing up my microcosm. The picture on the left is damselfly larvae, the middle is mosquito larvae, and to the right is scuds. These 3 were viewed under magnification on my phone so that I could snap pictures of them. Here we have pictures of the microcosm itself. Week 1, you can see that there is the green cattails in the top left. The algae had just started growing at the very top of the microcosm. And if you look right in the middle of that picture, you'll be able to see another heterotroph there, but I was unable to identify that one because I couldn't get a clear picture of it. And in week 1, you can see that the water was very murky. In week 4, the water cleared up. The algae, as you can see, grew from the soil level to the top of the microcosm. And then the top right of that jar, you can see where the cattail had died off. In week 7, you can see the algae is starting to die off. That cattail that you can see at week 4 has already decomposed into the water. My final findings from my microcosm were that in this experiment, it was found that with limited light and respiration, cattails will not survive. Algae can survive with limited light resources and will produce more of its own oxygen and carbon dioxide to support itself. The heterotrophs present in this environment can survive in very poor conditions and will even reach metamorphosis. My observations did not support my hypothesis due to the fact that my observations disprove my hypothesis because the cattail died long before the algae started to show any signs of distress. The 3 major course concepts that were evident for me in this project were the cattails need an ecosystem with proper respiration to survive. This project is relevant to how plants would react to a negative atmosphere, and this project is relevant to how aquatic heterotrophs are indicators of how much pollen there is present in an ecosystem. For general ecology, microcosm studies are important due to the fact that we can study an entire ecosystem or focus on just one key heterotroph or microorganism in a controlled environment. They can also be used to test the effects of toxins or pollutants on an entire ecosystem without damaging the ecosystem. For a future hypothesis, I'd like to test the survival rate of heterotrophs in a system that is devoid of algae to see if there's a higher survival rate. And if I were to repeat this project, this same one that we just did, I would remove the algae from the experiment and place another type of autotroph in the water to test the conditions. Listed here is my references for Google Maps so that you can find the area of Beatty that I collected from on your own.